Welcome, everyone, and thank you for joining the conference with Solid Biosciences. We are thrilled to be here talking with Ilan Ganot and his team about the recent results from the gene therapy meeting. With that, I'm going to turn it over to Eric to begin.
Hi, everyone. I'm Eric Camino. I'm the Vice President of Research and Clinical Innovation here at PPMD. I'm sure many of you saw yesterday that Solid shared an update of their 12 to 24-month safety and efficacy data from the first 6 patients dosed in their trial, and also shared that they have resumed dosing additional patients in their IGNITE DMD trial, which is their phase 1/2 trial with their investigational gene therapy product, SGT-001, which is delivering a microdystrophin transgene with AAV. So today we're going to be joined by members of the Solid Bioscience team to walk us through some of that data. So I'm joined on this call by Pat Furlong. And then from the Solid Bioscience team, we have Ilan Ganot, the co-founder and president and CEO of Solid Biosciences, Annie Ganot, the co-founder and head of patient advocacy, Dr. Catherine Clary, the acting chief medical officer, and Dr. Carl Morris, the chief scientific officer. So as a reminder, while we go through the presentation today, if you have questions, please feel free to put any of those questions in the chat box at the bottom of your screen. When we get to the Q&A session at the end, we will try to get to as many of those questions as possible. And with that, Annie, if you're ready, I'm going to hand the presentation over to you to get started today.
Thanks, Eric, and thanks so much to PPMD for hosting us today. We are grateful, of course, for the opportunity to speak directly with the patient community to provide an update on our IGNITE DMD clinical study, and we welcome the opportunity to take questions following our presentation. I know so many of you, but I'll quickly introduce myself. I'm Annie Ganot, co-founder, head of patient advocacy, and a Duchenne mom. I get the pleasure of speaking with Pat, Ryan, Eric, Kaylin, Rachel, and the rest of the PPMD crew on almost a daily basis. They make us a better company, and they help us ensure the voice of this community is incorporated into everything we do. Before we kick off, I would like to note that during this call, we will be making forward-looking statements as acknowledged on this slide. As most of you know, Ilan and I founded Solid after our son was diagnosed with Duchenne in 2012. We are based in Cambridge, Massachusetts. We are about 75-plus employees strong and focus solely on developing meaningful therapies for all patients living with Duchenne, regardless of age, mutation, ambulatory status, and disease progression. I would like to take a moment to acknowledge a member of our team we lost at the end of 2020, Michael Countryman. Michael was a beloved employee and family member at Solid for several years. Michael interned with us while he was finishing his bachelor's degree in communications at UMass Boston, and then he came to work full-time after he graduated. Michael worked in patient advocacy, communications, and he also oversaw our intern program. He himself inspired a whole new generation of young scientists. He was loved by everyone at Solid. This has been a huge loss, of course, for Michael's family, Judy, Ray, and Kevin, for our Solid family, for PPMD, and so many in our community. We are working hard to ensure that Michael's incredible impact and legacy lives on. I'm now going to over to Dr. Carl Morris, who will take us through our microdystrophin construct, SGT-001. Over to you, Carl.
Thanks, Annie. Hi, everyone. So I'm Carl Morris. I'm the Chief Scientific Officer here at Solid Bio. I'll just go back and review a little bit what we're trying to achieve here with AAV-mediated gene therapy. So as you're aware, there are 3 components in AAV gene therapy. First is the transgene, and for us, we have that SGT-001 microdystrophin. So I'll talk a little bit more on the next slide. The second part of it is a CK8 promoter. So this is a muscle-specific promoter that we put in there to ensure that we have high levels of expression, but it's specifically targeted towards the muscle only. So that's the cardiac, diaphragm, and skeletal muscle. And this was developed by Jeff Chamberlain and Steve Hauschka using in vitro and in vivo models, really to try to generate something that will produce a high level of protein expression. And finally, the capsid. The capsid itself, we have selected AAV9, which is one of the many serotypes that have been used clinically, and we selected that based on prior clinical experience from companies like Efficacy, and also Pfizer is using a similar capsid to us. And now moving on to the microdystrophin construct that we use in SGT-001. What I'm showing up the top is the full-length dystrophin protein in the long structure on the right-hand side. And the function of protein is really to stabilize the muscle membrane through connections at what we call the C-terminal end, so one of the ends, and also connection to the actin and the skeletal, the contractile system in the muscle itself. So that way it stabilizes the membrane, acts a little bit as a shock absorber, and prevents sort of that tearing of the muscle and damage. Unfortunately, Duchenne, this protein is missing, and so therefore there's contraction-induced damage that's associated with the disease progression. So what we selected in the SGT-001 microdystrophin construct, as shown down below, is that given the large size of the dystrophin protein, it's about 3 times too big in DNA to fit into the AAV. So we had to look at a rationally designed approach. And this came through many years of work by people like Jeff Chamberlain, who spent years iterating and identifying what was necessary to maintain a functional surrogate approach to the loss of the full-length dystrophin. Dongsheng Zhuang looked at the nNOS binding domain, and we thought to incorporate both of those into our construct would enable potential improved functionality, reduce some of the functional ischemia that's observed in boys with Duchenne, and give us a good functional surrogate to the missing dystrophin protein. And so, next slide. And what we were able to do through a lot of preclinical work, but now highlighted here on this slide in the clinic, and we've shown this previously, where if you're looking on the left-hand side, patients 4, 5, and 6 in our high-dose cohort, so receiving 2 E14 vector genomes per kilogram, we see 20 to 70%, an average of about 55% of widespread microdystrophin-positive fibers. And so what that's sort of telling us is the AAV is doing the right thing and going all over the muscle, so it's spreading throughout the muscle and giving us opportunity to sort of stabilize many of the muscle fiber membranes within the muscle tissue. Also importantly, and that really is sort of a functional approach, so a characteristic. So if you can get the protein to the membrane, so all those little red circles show that the protein is moving to the membrane, that shows that the protein is actually doing what it should be doing. In the middle panel, where we're looking at beta-sarcoglycan, so remember that's one of those membrane-associated proteins, the dystroglycan-associated complex protein. So what we can show here at day 90, so 3 months after we dosed with SGT-001, you can see that there's a co-localization of the microdystrophin and the beta-sarcoglycan, again indicating that our protein is functioning as we expected and being able to stabilize that protein complex at the membrane. And also importantly and uniquely to our construct is the nNOS binding domain. So on the right-hand panels, what we're highlighting here is in a similar section to shown, we are getting co-localization of the microdystrophin and the nNOS itself, which when you look at it, it provides us, again, a confirmation of protein function. So again, this is sort of important because it really sort of highlights the overall functionality of our protein. On the Western blot, and highlighted down the bottom there, is we do see about 10% of normal dystrophin using our Western blot, our qualified Western blot assay, with a range of 5 to 17.5%. So again, we're getting good expressions, and we look forward to moving forward. And with that, I'll turn it over to our Acting Chief Medical Officer, Catherine.
Thanks a lot, Carl. And I'm really pleased to be here with the Solid team and presenting to you at PPMD today. This is the first presentation since last night of our functional data for the first 6 patients that were dosed in IGNITE DMD. So next slide. So let's talk a little bit about the study itself. So IGNITE DMD was designed as a randomized controlled study, and after the first 3 control patients were entered and the patients were dosed at the low dose, we switched it to a non-randomized design. The intent of when IGNITE DMD was designed was to be able to treat both children and adolescents, regardless of age and regardless of genetic mutation. Because of some of the safety issues we've had, we are focusing now on the younger age group. We do recognize that there's a great need in middle ages and the older adolescents, and certainly we would love to get back to treating that population at some point. We have these patients have to be stable in their cardiac and pulmonary function. They need to be stable on a daily dose of corticosteroids for 12 weeks before entering into the trial. And we do have 4 open sites, and University of Florida, where Dr. Byrne, our principal investigator, is located, and he actually dosed the first 6 patients. Dr. Perry Shea at UCLA is a recently opened site, and he dosed our 7th patient, who was dosed with our new manufacturing process and new risk mitigation protocol. And then UMass and Lurie Children's Hospital are also open sites. Next slide. And this shows you the 3 groups that we're going to be looking at as we walk through the data. So I mentioned that there was a control cohort, and then there's a low-dose cohort, 5 times 10 to the 13 vector genome per kilogram was the dose, and then the high-dose cohort that Carl mentioned, 2 times 10 to the 14 vector genome per kilogram. So we'll refer to these groups as CT control cohort, patient 1, 2, and 3, and the low dose patient 4, 5, and 6, and the high dose, just to orient you. And as you can see, if you look at the ages across, there's quite a variation in age. Unlike some of the other gene therapy studies that have been done where there's concentrating on a lower age group, our commitment again was always to treat the older patients as well. So what you see is that there are 2 adolescent patients, one in the control group, one in the low-dose group, and of course they were not able to perform the North Star ambulatory assessment of the 6-minute walk test because they were non-ambulatory at the time of study entry. Next slide. Before we sort of do a deeper dive into all the various assessments that we'll be presenting today, I wanted to offer or show you this table, because this table is really a snapshot across all 3 of the groups of the different assessments that we have performed. So in the control group, it's the top, the low dose group is at the middle, and in the bottom, you see the high dose group. Red indicates a worsening, green indicates an improvement. And as your eyes just scan across and down, what you see is that in the control group at the top, there's unfortunately worsening in many of these parameters as opposed to improvement. When you look at the middle column, which is the low dose group, you begin to see more improvements than worsening. And as you look at the high dose group, literally almost every parameter is green, showing improvement. Because we know that Duchenne is a very complex disease with lots of different outcomes that occur both in skeletal muscle and the pulmonary system, the cardiovascular system, how a patient feels and functions. The intent in designing IGNITE DMD was really to try to assess a lot of these various outcomes. And the other thing is, because the study was designed in order to accommodate both younger and older patients, we felt we needed to provide a variety of assessments. So that's why you see this broad range of assessments here. And I'll go into each of these specifically in the next slide. But we're encouraged by this slide because we do feel that it demonstrates that there are improvements seen more in both of the dosing groups than the control group, and certainly with a dose effect where you see more improvements in the high dose group. Next slide. So these are the various endpoints that we're going to be looking at. This is a first in human trial, so of course we're looking at safety, which is very important, the most common adverse reactions and lab abnormalities. Carl had already talked about the biomarkers. Certainly microdystrophin protein expression is our primary efficacy measure. Creatine kinase, which as you know is a protein that is released from muscle, is also a biomarker that we're utilizing. For functional assessments, what you're going to be seeing today is the North Star Ambulatory Assessment, the 6-minute walk test, and a pulmonary function test called forced vital capacity. And then we feel that it's very important to understand from a patient and parent perspective how they feel that the patient is functioning. And so you're going to be seeing some patient-reported outcomes. This, to our knowledge, is the first gene therapy trial that has actually shown 6-minute walk test and patient-reported outcomes and pulmonary function testing. Next slide. So, safety. First of all, what we saw is the most common drug-related adverse reactions were nausea, fever, and vomiting. And you've probably seen in other trials, these are very common after the infusion, of the gene therapy infusion. They're generally transient. They last for a couple of days, and they can be treated. So this is almost all the patients had these symptoms. And I should note that also patient 7, who we recently dosed, and we talked about him last night, had the same symptoms, which were transient and quite manageable. This slide also shows the most common laboratory abnormalities, and one thing of note in our trial is that every patient did have drops in their platelet count. This seems to go with the territory, once again, with gene therapy treatment. What's encouraging is there are no clinical symptoms. It's a laboratory finding, really. There's no clinical symptoms associated with it. And all patients did have laboratory evidence of complement activation, but there were 3 of the 6 patients did have 4 SAEs. The positive thing, I think, is that the SAEs occur early on in treatment. We now have learned how to manage them and utilize those management techniques with patient 7. And after the patients get to that very early few weeks after the gene therapy infusion, we have seen no new drug-related safety findings. And we've been following our patients now for 17 to 37 months. Actually, the first patient in the trial, just very recently had his 3-year visit. So after those initial couple of weeks, they get through that period of time, and then they at least in our trial, are doing very well. Next slide. Now, I mentioned creatine kinase. It is a very variable measure. A boy who's walking better or riding his bike can go out and have a very active day, and then his CK levels will increase. But what is meaningful to us on this slide, while there certainly is a lot of variability, is to look at the right-hand column. And if you see the high-dose patients had a 55% decrease with a low standard deviation of 7.5%. So we feel there's, if you look at this in the context of microdystrophin expression and the functional outcomes, it's suggestive that there may be some membrane stabilization going on. Next slide. Now I'm going to move on to the functional outcomes data, and it's really important. Again, remember that our patients were quite variable in their age, and there were patients entering the trial who were older. This slide actually shows what we would see in natural history with Duchenne. So the North Star Ambulatory Assessment graph on the right is from the UK database. It's 395 patients. Each of those gray trajectories represents one patient. And of course, you see a lot of variability there. It's very heterogeneous disease. The mean is that black line right in the middle. So that's sort of the mean. In general, and as a mean, what we see is that boys with Duchenne from the ages of 4 to about 6.5 or 7 are increasing around 2 points a year on the North Star Ambulatory Assessment. That's what we would expect to see. On average, after the age of 7, there's a decline of about 3.7 per year. So if you see a stable core at the age of 10, when you were expecting to see some decline, an average decline, that is positive. So next slide. Now let's look at our data on the superimposed on the graph, the natural history graph. And what you see here is that the high-dose patients are in blue, the low-dose patients are in purple, and those control patients who have not been treated are in black. And on the left is the baseline to 1 year graph. And what you see is that the 3 high-dose patients are improving. One of them has dropped 1 point, and the control patients are declining. Then if you look at baseline to last visit, you see that, once again, high dose in blue, low dose in purple. You see that the patient who had been declining in the low dose group has begun improving. And actually what's interesting here is we followed the low dose patients now for 2 years. And we find this to be an intriguing finding because the primary efficacy endpoint for gene therapy trials has typically been 1 year of follow-up, but it could be that there, not only that is the improvement sustained, but there might be further improvement over time, and this is something we do want to follow up. So what we're seeing basically is that our patients had rather high baselines. They improved versus the natural history, and they also improved more so than the control group. You see the control group here on the left in gray, and then the high dose group, the low dose group, you see that additional improvement at 2 years. Next slide. 6-minute walk test as well shows a decline in natural history, and this is the natural history slide, of about 35 to 50 meters in distance per year, beginning around the age of 7, and that's what this slide shows. Next slide. Here again, we see the IGNITE DMD 6-minute walk test overlaid on a large natural history data set that highlights the mean natural history trajectory. So you see the individual scores, you see the mean, and actually those dotted lines represent the variability around the mean. And on the 6-minute walk test, we saw in both low and high dose groups, they had absolute improvements in their 6-minute walk test distance from baseline to 1 year, with the mean changes above the generally accepted minimally clinically important difference of 30 meters. As shown in the figure on the right, the 2 low-dose patients had further increases at 24 months. So we were seeing that both in North Star and in the 6-minute walk test. Next slide. Here we see the individual 6-minute walk test distances for each of the patients at 1 year and at their last visit. And you see the mean change up the top is kind of small, but the mean change for each dose cohort was greater than that 30-meter clinically meaningful distance that I mentioned, versus really no change seen in the controlled patients. So individually, both of the low-dose patients and the 2 younger high-dose patients showed that clinically meaningful change of over 30 meters at the last time point assessed. So the results on the 6-minute walk test and the North Star, we feel represent several different aspects of patients' physical functioning and also of their ability to partake in daily activities. But another really important parameter is pulmonary function. And as you know, as boys get older with DMD, they begin to have pulmonary deterioration due to the weakening of the diaphragmatic and accessory muscles, and this causes a restrictive pulmonary disease. This slide shows that there is, on the left, there's deterioration that we see in natural history that starts even really before there's clinical symptomatology when compared to, age, sex match and weight match controls. So they're starting to deteriorate, but then you begin to really see that functional deterioration that's noticeable in middle childhood to early adolescence. On the right here, we show a similar type of graph where the high-dose patients are in blue, the low-dose in purple, and what you see is improvements over our control groups, which are declining, and improvements also over what you would expect from natural history, which is seen on the left, what's interesting is even the very young boys were improving in our trial on this forced vital capacity, which is a very good spirometry-based assessment measure in Duchenne. So we're very encouraged by the variety of these functional endpoints, and we look forward to developing more data. Again, 3 patients in our high dose group and 3 patients in the low dose group is it's a very small sample size, but we do feel it's encouraging that we've seen these various outcome measures. Next slide. And this just shows the change scores in the forced vital capacity. And it shows absolute change from baseline for the percent predicted FVC. And you see the control group declining, the low dose group showing some improvement with one patient having a slight decline, and then improvements seen in the high dose group. Now I'd like to turn to the patient-reported outcomes that are being evaluated in the IGNITE DMD. And again, we're really excited to be able to present these outcomes as they haven't been presented before in a gene therapy trial. Next slide. So we're actually going to be showing you 2 different patient-reported outcome assessments. One is the Pediatric Outcomes Data Collection Instrument, also called the POD-C. And the POD-C is a well-validated scale that's been utilized in orthopedic and rheumatologic and neuromuscular clinics for quite some time. So it's really well understood. And it has 6 subscales. It has sports and physical functioning, transfer and basic mobility, upper extremity and physical functioning, and then happiness and pain, and then global functioning, which really is representative of the entire compendium of all the different subscales. Modus outcomes research, we worked with this outcomes group to develop a specific semi-structured qualitative interview for Duchenne that is conducted with the patients and their parents at a year, well, at baseline and then at a year. And it's a proprietary guide that was developed for us. And the symptoms that are assessed are pain, fatigue, and stature. And then the impacts, and these are the functional impacts, again, as assessed by the patients and their parents on activity, school, social, psychological, and functional. Okay, the next slide. And as shown here, patients with Duchenne have a steady decline in their motor function compared with healthy children of a similar age, as measured by transfer and basic mobility domain, the POD-C, and the sports and physical functioning domain of the POD-C. It's interesting that there is a good correlation that's been seen between the POD-C global subscale and transfer mobility scores and the changes in the 6-minute walk test. So we find that actually to be quite validating, that the patients and their parents and their assessment of how they're functioning is well correlated with the 6-minute walk test. Overall, we feel this is an encouraging outcome. And if you look at the change scores here again, it's easy to see the dose-related response, where the control groups are declining in the global scores, sports, transfer, and upper limb domains. The low dose cohort is improving on most domains, and then the high dose cohort is improving on all domains. Next slide. Now, I've mentioned the Modus outcomes research, and this just is more of a representative of all the different aspects that you see under activity, school, and psychological. And activities, obviously, are activities of daily living. You see them here. They're things that are done every day in school, and social, which is really important. Feeling different, left out by peers, keeping up with peers or siblings. There's a psychological aspect to it and a functional aspect to it, which involves motor weakness, transferring lower limb mobility and upper limb mobility, very important aspects of motor function. And the way we're going to show the Modus outcomes research scale and changes seen in IGNITE DMD is that if you see a green box, it represents improvement or positive change. If you see a red box, it indicates worsening or decline, and then a white box is just stable or no change. And this is the results from the Modus outcomes, and it shows the symptoms and impacts in the study. First of all, on the left, you see the control group. Unfortunately, most of the boxes are red, as you see. There's one green box, but unfortunately, most of the aspects were worsening. In the low-dose cohort and the high-dose cohort, you see that there were no worsenings at all. There was a lot of boxes that remained stable, but there were many green as well that were seen as improved by the parents and the boys. And if you look at the high dose, you see that there's many more green. And one of the, can't see very well on the slide, which is why I showed the previous slide, but there's several aspects in the high dose group that we saw that all three patients in the high dose group, as well as one in the low dose group, had improvements in the symptom of fatigue. And we feel that that's supportive of a potential effect of the nNOS binding domain that Carl talked about as part of the microdystrophin construct in SGT-001. So we felt that the results here were very promising. They're certainly consistent with improvement in motor function domains that we saw in the treated patients, and agree well with the POD-C results that I just showed. Now I'll hand it back to Carl for a review of the previously reported IGNITE DMD clinical protocol updates and solid second-generation manufacturing improvements. Carl?
Thanks, Catherine. As we reported previously in October 2020, the FDA lifted the clinical hold on IGNITE DMD, our clinical study. Since then, we made several, to get off hold, we made several amendments to that protocol to enhance the patient safety based on our understanding of what actually caused the serious adverse events in the prior patients. So another important amendment was actually to reduce the total viral load to a second-generation process, which I'll talk about in a second. What we're highlighting here really is we took a three-pronged approach to this, and it's first was to inhibit the complement activation that we are observing, and we used two different inhibitors. One, eculizumab, which you may have heard of before, which sort of treats the end part of the activation of the complement system, and another one called C1 esterase inhibitor, which treats the first part. So it actually tries to stop the initial activation. The second thing we've done is we actually increased the steroid dose from what we were using previously at 1 mg/kg to a very transient increase up to 2 mg/kg, just to manage that initial acute time frame where we saw the issues previously. And then finally, with the first two patients, so that's patient 7 and patient 8 only, we agreed to reduce the limit to 18 kilograms in those two patients. And after that, we can now move forward up to about 30 kilograms. Next slide. So here's what we call a second-generation manufacturing process. So previously, what we had, and shown on the left-hand side, is an electron microscopy image of a pseudo-coloring that shows red and red full capsids. So those that contain the construct, the DNA that we want to deliver to the muscle cells. And the blue ones are actually empty. They did not contain anything. It was just sort of empty capsids. So imagine that as delivery trucks. One has the cargo, one doesn't. So we wanted to get rid of those that did not have the cargo. So you can see with our new process, we were able to actually increase and enrich for the red, the cargo-containing delivery vehicles, and therefore improve to 90% full capsids. And what that's done is really sort of highlighted on the right-hand side. While we maintain our dose, so we dose with our vector genome, so how much DNA we're delivering per kilogram, we've actually been able to reduce the number of capsids per kilogram, so and significantly reduce that 47%, so about 50%. And this is expected to also provide some improvements in the safety profile of SGT-001. Next slide. So as we went, and this is what patient 7 was dosed with. So patient 7 was dosed with our new second generation process material. And what we were able to do prior to actually dosing, we established some rules. It's called a priori, but it's just before, to determine the effectiveness of the risk mitigation. And really what it had 3 components. The first was the absence of any serious adverse events, which we achieved. Also, the absence of complement-mediated adverse events. So anything that we considered linked to that complement activation was seen previously, and also that was achieved. And then finally, we wanted to make sure that we were actually substantially reducing the complement activation. So this is a laboratory measure. So we take the blood and we measure certain components of the complement system. And we wanted to make sure that our complement inhibitors, as well as the reduction in total viral load, actually showed improvements. And that's what happened. So we are very, very happy with the safe dosing that we observed with patient 7, and truly believe that our clinical mitigation strategy, as well as our second generation process, has sort of helped us. Clearly, one patient, we need to do this many more times, but we are happy with what happened here. And now I'll turn it over to the CEO, Ilan Ganot, for closing remarks. Ilan?
Thank you, Carl. And thanks, everybody, for dialing in. And thanks, Pat, and the whole team at PPMD for having us today. For me, it's a first. As Annie mentioned at the beginning of the call, for us, it's a very personal mission. We have a 10-year-old son with Duchenne at home, actually in the back room right behind us now. You know, from the beginning, we knew we couldn't be cutting corners, and we had to hire the best talent in order to do this right. This data and what we just shared with you, for us, is a combination of 7+ years of learning, preclinical studies, advancements made in the clinic, and a lot of improvements to manufacturing and process development to support this development of a clinical candidate for our boys. You know, it's not been an easy journey or straightforward, but I am proud of the progress that we made to date, and really excited for the future of the company and for this construct, SGT-001. We're pleased to see promising evidence of benefit in the 6-minute walk test, in the North Star Ambulatory Assessment total score, all following the administration of SGT-001. The improvements to the pulmonary function test, as well as clinically meaningful improvements in patient-reported outcomes that assess motor function and fatigue are really exciting. We take them together and we look at the totality of the data as something that potentially supports the role that our microdystrophin is playing in providing a differentiated benefit to the patients that we've treated, and hopefully to the many more that we'll treat in the future. We are very glad to have safely resumed dosing. Those of you that know me know that those safety challenges that we've experienced were, you know, amongst the worst experiences in our life, that's for sure. And it is great that this patient we just treated, and hopefully everybody we treat from now on, will be a lot safer. And that's thanks to a lot of the changes that we made to the protocol that Catherine's reviewed, as well as some of the manufacturing improvements that we made to the drug product that Carl just highlighted. So really, on behalf of everyone at Solid, I'm thankful to the FDA. They've been a good counterpart for us. We've had a good and helpful dialogue with them that ended up in us lifting the clinical hold and resuming dosing just now. I want to thank everybody at UCLA who treated our 7th and recent patient, Perry Shea, and his team, as well as, of course, everybody who is enrolled and participating in IGNITE DMD. We recognize how challenging it is to be a part of the clinical trial, and really grateful for everybody who's taking part. We couldn't do this without you. This is a journey that we've taken to try to really improve the outcomes for patients living with Duchenne. And I look forward to updating more data and more outcomes in the future and hopefully see everybody, or have everybody hear us, or hopefully even in person in the next PPMD annual conference in June. So, we'd love to take some questions, and Pat, Eric, over to you.
Yeah, thank you. Thank you so much, Ilan, and thank you, Carl, and thank you, Catherine, and thank you, Annie, before that, and thank you all. This has been a lot of information, so if you don't mind, I'd like to go back to and start at slide 7. And I know this was over to you, Carl, so I just want to go through them a little slowly. This was a lot of information, and certainly what we see we like. So I'd just like to go over some of the questions we received and some of the questions that I've thought of along the way, if you don't mind. So, Carl, on slide 7, you talked about the binding sites, and I think this is a really hard concept for people to understand. So when you talk about the binding, you're talking about the binding to different parts of the cell, to be fair, and you've talked about the reason that you all included the NOS binding domain because of ischemia. Could you just give us a few explanations of ischemia and what that does when cells are ischemic, and why NOS is there, and what talk to us about the binding, and talk to us about NOS and ischemia and what that means and why you wanted it in there.
Okay, sure, Pat. So I think first, remember that the binding is at each end, and so that's really to stabilize the membrane with the cytoskeleton. So the inside, the actinomycin that causes contraction, and so we actually could lift things and walk. So we have to connect that with the outside. So that's where, if you look down at our construct in the bottom right, the orange box, and then on the left-hand side, and then the gray box on the right-hand side. So the orange connects to the inside, and the gray connects to the outside. And those domains are taken directly from the full-length construct that you can see up in the top. Again, the orange on the left, and then the gray right next to the other orange on the right-hand side, connecting to what is BDG, which is beta-dystroglycan. So those are the outside. But as you can see in the full length, there are also other domains within the microdystrophin, within the full length dystrophin that bind all those little circles with R, 1 through 24. A lot of them actually can bind at different proteins along the length of the construct. And we're highlighting up in the top that nNOS, which is neuronal nitric oxide synthase, that's an enzyme that normally just connects to the dystrophin. What that does is it actually generates nitric oxide. Nitric oxide then, in turn, is an activator of vasodilation. So when everyone starts moving around a little bit more, there's sort of a call for more blood flow to the muscle. And so, and the blood flow to the muscle is increased so you can get oxygen into the muscle. So when oxygen's in the muscle can function and behave better. So if it doesn't have oxygen is called ischemia. And so what we're hopeful is that by including the nNOS binding domain, we allow for the functional surrogate of nNOS binding to that domain, allowing for NO to be produced appropriately, improving blood flow and oxygenation back to the muscle. So when the boys are moving around, then they'll have the right environment to continue moving around. So that's why we talk about fatigue and this ischemia. Does that help?
Yes, Carl, that's very helpful. I think that just understanding ischemia and the fact that you're trying to deliver more oxygen so the muscle operates better or has better circulation to it is very important. And then if we look at slide 8, tell us about what muscle you were looking at. So this is really terrific that you have between 5 and 17.5% of the dystrophin construct there. Tell us what muscle was used in the biopsy.
So we use the vastus lateralis, which is actually in the thigh. So in the front of the thigh is the muscle that we use.
I think that's probably a very good muscle because it's a large muscle, right? So that you can see distribution there. So that's a very good sign. All right. Thank you, Carl. I'm going to go over to Catherine now.
Okay.
Mm-hmm. Catherine, if we go to slide 10. So you talked about switching from to a non-randomized design. I think that's a hard concept. Could you just tell us a little bit about non-randomized design and what you switched to the non-randomized design?
Sure. Yes, when we originally started the study, we randomized patients, meaning that they were allocated to one group or another, and the two groups were to be treated with the SGT-001 right away, or to wait a year, and then they would be dosed at the end of the 1 year. So that was in the first part of the study, and we'd actually planned to have that continue as a study, and then we would be dosing the patients after 1 year of waiting, and they would serve as sort of a natural history control that we could compare the dosed patients to. However, as we had some SAEs and decided that it was necessary working with our DSMB to decrease the weight in the study, we did change that design such that patients are now just entered into our dose, which is 2E14, and they're no longer randomized to that delayed treatment control group for a year. I do want to mention that we're very grateful to all the patients who are participating in the trial, but in particular, certainly the control patients, because they continue to hang in there. Dr. Byrne remains in contact with them. They continue to come in for evaluations, and we're very hopeful that we may be able to dose these patients in the future.
Thank you, Catherine. That's very helpful. So as the study is, sorry?
Yeah, sorry, just to go back to the biopsy. We go on the slide, and we're looking to use needle biopsy wherever possible. I just wanted to make sure you're aware of that as well.
Yes, thank you, Carl. I think that every parent is hopeful for no biopsy, but under these circumstances, recognizing the importance and necessity of biopsies. So we all prefer a needle biopsy. That is a biopsy that's taken without using general anesthesia, and is certainly a less invasive type of biopsy than an open biopsy. So thank you for that. Catherine, just continuing on this particular trial, how many individuals are you going to include in this trial going forward? Do you have a number that you're going to reach, or is it open in terms of you're able to expand? So how many are you thinking?
It's a great question, Pat, and the trial design is quite flexible. It allows us to dose anywhere between 16 and 32 total patients. And of course, we've now dosed 7. Our thinking is, as we dose patients this year, we're certainly learning about the effectiveness of our safety profile, and we also, of course, want to see the efficacy in the patients that we're dosing under the new process and the new protocol. So we'll be dosing this year, and we are starting to think about our registration trial. We haven't set on a number yet that will be enough in order for us to approach FDA and other regulatory authorities about that registration trial, but it certainly will not be at the high end of the 32 patients. It'll be more at the lower end or even a little bit lower.
Right. So when you're talking about a registration trial, you're talking about a different trial than is currently active, and you're talking it would be considered a phase 3 for a confirmatory study with the goal of getting an approval in the space. Is that correct?
That's exactly right. And we, of course, would like to get as much data as we can, as quickly as we can, so that we can get to that trial and we can get the drug to patients.
Sure, of course, and I think we all wish you well on that. So you'll be using the 2E to the 14th dose in this ongoing trial and then a registration trial. Is that right?
Yes, and that certainly is the plan in all likelihood. Yes. I mean, we have no immediate plans to increase the dose any further.
Great. And one of the questions.
We just started dosing. We've just started dosing with our second generation process. And so, we'll dose a few patients, and we'll have the biopsies and evaluate the protein expression and look at sort of early functionality of that, and then we can make a more informed decision about long-term.
Sure. So you've dosed your 7th patient. Do you have? I'm sure you do have plans, but are you able to tell us within the next few weeks you'll dose patient number 8, and then that limitation in terms of weight will be lifted? So can you shed a little light on that?
Sure. So we're very actively screening patients for that next patient, and we do expect to dose within the next few months. As you know, our protocol, we do have to go back to the DSMB and want to after 30 days of clinical and laboratory assessments after each of those first 2 patients, which could be only 18 kilograms or less. Our protocol, though, allows us to go up to 30 kilograms. So assuming that we safely dose the next patient, the plan would be to begin to incrementally increase the weight of the patient so we can get into that, once again, begin to approach that broader population that has always been our objective.
Right. Thank you, Catherine. So basically this next patient will be under that 18 kilograms. Then you go back to the DSM, to the IRBs and to the FDA, and then get the agreement to go beyond that weight limitation, right?
Yeah. Let me just clarify.
Sorry?
We don't have to go back to the FDA. We just will go back to our DSMB. Yeah, the FDA approved our protocol and the knowledge that we'll be talking to DSMB about the first 2 patients dosed, and then we will be able to start increasing the weight.
Great. So the DSMB, for those of you on the line, is the Data Safety Monitoring Board, and this is a group of individuals that Solid will go back to confirm the safety of patients 7 and 8, and then to expand beyond that weight limitation. I don't know if you're allowed to say this out loud, but there are obvious questions in twofold about what, in terms of your neutralizing antibodies, are you able to tell us what the cutoff is, and do you have strategies to get around people who screen fail for neutralizing antibodies?
So I think we use an antibody assessment, and we can't, even communicating the number wouldn't really, it's assay dependent. So the number, we haven't disclosed that, and so we can't. But I think we've identified something where we think is an appropriate level. And clearly in the background, we're looking at multiple ways to get into the patients that are what is called seropositive. So those that have antibodies against, right now, AAV9. So yeah, we're actively looking into other approaches to get around that issue, to reduce the antibody levels for those that don't meet the inclusion criteria on our total antibody assay.
Right, Carl, but for this current study and the registration study that you're planning, those strategies would not be included for patients who screen fail based on antibodies. Is that correct?
Yeah, that's correct. It would need to be a second or an additional study.
Right, it would have to be a different study to think about that, right?
Correct, that's right, Pat.
Okay. All right. Thank you. And just to reiterate, you're going in with a high dose, so no one will be in your studies and ongoing studies will be given a lower dose than the 2e to the 14th. It'll be consistent now. So it's not like you're using 2 different doses.
We are not using 2 different doses. We've just removed the empty trucks, the empty capsids.
Yep.
So the virus the amount of virus that we're adding is reduced, but the amount of vector genomes, so the amount of DNA copies is the same at 2e14 vector genomes per kilogram. That's right.
Good. So I'm going to go to slide 14 now, if you don't mind. So this is where we've talked about the side effects and the complement activation, and this is where you began talking about that. And I think the nausea seems pretty consistent, at least anecdotally, people are talking about that. So, Carl, maybe you can shed some light on this. At what point in time following an infusion do you see the complement activation or the decrease in platelets? You then start a mitigation strategy of giving medicines to this individual, and then how long are the medicines given, and how long does this when do you see the platelets begin to return to normal?
So I'll start, and then Catherine, obviously can step in. I think all of these, as we mentioned, the transient, we see very, very early on. We are going with a pre-treatment strategy with the complement inhibitors right now. So we are actually pre-treating prior to SGT-001 dosing to make sure that we're reducing the complement activation. I'll turn it over to Catherine to talk through some of the kinetics of it all.
Yeah. So, Pat, not only with SGT-001, but with other gene therapy products, the drop in platelets is seen, and it usually occurs with a pretty consistent pattern of around 4 days after the gene therapy infusion, and it drops by pretty significant amounts. In our case, it got below what's considered a quote-unquote normal range for for platelets. What is very interesting, though, is that there are no symptoms associated with it. So normally when you get to a very low platelet level, you'll see some signs of bleeding. You might see some, and we don't see that at all, no matter how low the platelets go. And then after a few days, it comes back up, and by 2 weeks, generally it's way back up to the normal level. That's a very consistent pattern. In our 7th patient, who we pre-treated with the complement inhibitors, as Carl mentioned, what we saw was an earlier, the drop started at around the same time, didn't go down as low as our other high-dose patients, and rebounded faster. But again, no clinical symptoms at all associated with that thrombocytopenia. In terms of the complement reductions, we see reductions in what's called C3 and C4, which are sort of the early phases of the complement pathway, starting around day 5, 6, 7. And then what happened in our patients who did not get pretreatment was the formation of what's called C5b9, which is also known as the membrane attack complex. It's an apt name because what that actually does is it attacks endothelial cells within the body, and that's what causes the complement-mediated adverse events. Again, with our patient 7, who we pre-treated with the 2 complement inhibitors, we saw a little bit of those complement laboratory changes that I mentioned, much less than any of our other 6 patients, including the ones who had no SAEs, and everything returned to normal much faster on a laboratory basis.
Okay, so Catherine, thank you. But I'm going to take that apart a little just to explain it. So first of all, this individual that was treated, patient number 7, was on 2 milligrams per kilogram of prednisone. Is that correct? Let's start there.
That's correct. Yep,
okay.
That's correct.
So then you pre-treated him with these drugs to prevent these things from occurring, correct?
That's right.
Okay, and so that they didn't occur. And you're drawing blood on these individuals frequently, daily, I would suspect in this case, to look at those platelets to see when they dropped or how far they dropped and to make sure that they were returning to normal. Is that correct?
Yes, for the first 2 weeks, we do require blood work. Sometimes, a line can be used and inserted so that you can draw the blood from that line, so it's not, drawing blood over and over.
It's not invasive. No, and I appreciate that. It's not invasive and stick every day, but you do labs every day so that you're really closely monitoring. Yeah, so that was the point. You're closely monitoring.
Monitor very carefully.
Right, because people don't have symptoms, so there has to be a way for you to understand when that platelet count is dropping and how to watch it return to normal. So I think that's really important to understand because as we all know, this immune system and the complement activation and all this is pretty complicated stuff. And so the whole point of this is you're on 2 milligrams per kilogram of prednisone. You've pre-treated with these drugs that you've mentioned, and then really monitored the blood work to make sure that the changes that you see were less than typical in other cases and came right back up to normal. And this occurs quite quickly. It's not going to occur 6 months later. It's going to occur quite quickly so that you're monitoring it. All right.
Absolutely correct.
Okay. So then let's see. The natural history on slide 16. So these are just questions about how long are you going to monitor these children? And I think that the FDA is requiring 5 years. Is that the same that you're going to do monitoring these individuals frequently over the first year, clearly, and then it changes a bit, but you're going to monitor for 5 years out? Is that right?
Yes, our protocol does involve monitoring for 5 years. Yep. Okay,
that's fine. And one of the things I think that's on everyone's mind as we think through, and it's not a question, it's just a plea, if you will, that, we really need to look at holistically at these kids and think about their lives as well going forward in gene therapies where we could potentially look at combination therapy. So just a statement, not a question. Slide 19, please. Catherine, we're always mentioning MCID, and I think that a minimal clinically important difference. And so could you just give a little bit of insight into MCID so those on the phone can understand what you're looking for there?
Yeah. So previously, you're highlighting the 6-minute walk distance, and it's been used before by company PTC with the Adeluro and now Translarna drugs. So they really were at the forefront of identifying this and worked with Craig McDonald and many others to determine what is a meaningful change in how far the patient can walk in 6 minutes. And that was identified as about 30 meters distance in total. What you can see here is that there is a decline, starting at about age 7, and this was sort of identified both in this review paper by Kate Bushby, as well as sort of in the imaging DMD cohorts. They do see a drop, as you actually get older, past that 7 years of age, and it's about 35 meters to 50 meters. And so really what we're trying to highlight here with some of the data that we shared today is that we're seeing changes that look promising over 1 year and then also a 2-year period where we're above that what is sort of defined by regulators or accepted by regulators as a minimally clinically important difference, and that is that 30 meter difference here. And where we're seeing on average in the high dose cohort about 50 meters, and then the low dose cohort just about 40 meters in 1 year, but all 2 years is even significantly greater.
Right. Thank you, Carl. And the MCID is important to regulators as they review the data and make decisions about approvals or not. I think that's really important, and clearly on slide 22 and 23, where we saw improvements across all parameters, especially in the high dose, we really think that's terrific news for all of us. Clearly, more data is needed, but as you expand this study up to the number of patients in the 30s, we should see more compelling data that we're very hopeful to see in the future. Slide 25. Catherine, I think that we're familiar with the PODCI measuring all sorts of different domains of the patients, and I think that making sure you collect patient-reported outcomes is really critical because, it's one thing to have a patient respond to a number of outcome measures, the North Star, the PUL, the forced vital capacity, 6-minute walk, and we can go on, but it really matters how it happens. What real life looks like, right? What the days look like at home, what the days at schools look like. So we do appreciate the fact that you're looking at the PODCI and the Modus, because I think that really does say, here's what we're seeing in the clinic with the outcome measures. Here's what we're seeing with the dystrophin expression. Here's what we're trying to mitigate. We're trying to take care of this little one and this young man as he grows. But now we want to make sure that within his home environment, things are the same and pointing in the right direction. So we do appreciate all of those domains and continued improvement, hopefully. I think that brings hope to all of us. And now slide 30, and I know we're a little bit over time, so I'll wrap this up quite quickly. Slide 30 in the manufacturer. First of all, thank you for explaining that, Carl. It's really hard to understand that when you're delivering a virus, sometimes one of the issues is that sometimes the virus doesn't have the promoter and the microdystrophin in it. Those are called empty capsids. So decreasing those empty capsids so the amount of virus that an individual is getting is filled with all the stuff that is going to be beneficial to muscle, I think is really, really important. Some of the questions around this are about weight. Is there a weight limit when you're thinking about delivering 2E to the 14th virus as you're thinking going forward? People are asking, and I know I didn't see a weight limit. I saw a weight limit at about 66 pounds or 30 kilograms. Are you thinking ahead of that and going beyond that weight based on good manufacturing process and your efforts?
Yeah, we're very happy with what we've been able to do with the second generation process here and get rid of those empties, get rid of those things that are unnecessary. So that's sort of the first part. The other thing is we're now sort of going to incrementally and sort of in an appropriate way sort of work our way towards sort of expanding that total viral load. Ultimately, we wanted, and that's the mission of here at Solid is to dose, all patients, try and help all patients with Duchenne. So that means that there are going to be boys and young men who are all greater than 30 kilograms. So we want to do that, but we want to do it thoughtfully and appropriately and sort of work our way and make sure that we fully understand, or as best as possible, understand the safety issues and move forward in that way. So yeah, we want to get there. Right now we're at 30 kilograms per protocol in IGNITE DMD. But again, a second study, an additional study with no weight limit could be a possibility, and also just changing our protocol, and ultimately in the future might be in order.
Great, thank you. And now some general questions, and then we will wrap up within about the next 3 minutes. And I can't thank you enough for all of your time today, all of the people on the phone. I wish we were in person, but hopefully someday soon. A couple things.
Someday.
And this is just up for grabs for anyone. International trials. What are you thinking? When are you thinking?
Yeah, I mean, Pat, we definitely are thinking of a global trial for our registration trial. So,
yeah.
And so be for the registration trial.
Great. And okay, now this is a crystal ball question. And so all the caveats around crystal balls, that they're not always perfectly in their accuracy. They're about wishes and hopes. Can you give us some of your best guesses on a registration trial? What year? I won't say what month. I'll say what year.
Yeah, Pal, we safely dosed patient 7, this quarter. We have to take it as, I know it's not the best thing to hear. We have to take a couple of baby steps first before we can look into it. It's an obvious next step. It's something that we really have to push hard to get to. And trust me, Alon and Annie will not let us sort of dilly-dally towards that registration study. So we are actively working on various things here internally in thinking about what we need to do. We're pulling together the data we have, and I think IGNITE, in total, it has really sort of given us a really good understanding of what's out there. So we're dosing patients from 4 to 7, up to, we have the ability up to 17, but 4 to 11 in the ambulant population. And we have all these different endpoints, which, as you know, can better assess the different subgroups in terms of age. And so we're collecting those data. We're now up to 2 years for the low dose, and moving forward into 2 years in the high dose as well. We're going to collect more data and really assess those, look at the natural history data, go to the KOLs, and really be thoughtful about what we need to do to have the smallest and quickest next study as possible. So I mean that's really the goal, and hopefully we can get there sooner than later.
Great. And your older patients, they too generally improved, right? The older boys at the higher dose, or the older boy in the higher dose, was generally improving.
Yeah. Versus natural history and also absolute in terms of 6-minute walk, and as well as the pulmonary function. Yes.
Yeah, thank you. Okay, now here's the last question. I know that we are all interested in an approval. If we all cross our fingers and pray to whoever we pray to, do you think that we might see an approval in 2023? And again, we won't hold you to it, but we will ask it again and again. Alon, I'm going to just ask you.
Yeah, I'll take it. So first of all, I think to answer your first question about the when is the pivotal study, it's not just when, it's also what does it look like, and how soon before when we started can you finish it and therefore file with it, and so I won't give you a long answer like Carl. I'll just say next year.
Okay.
For your first question, on your second question, it says, are they going to be the year after that or the year after that? Because if we are going to be forced into a placebo-controlled design, it's going to take longer. But maybe with the help of people like you and others on the line, we can find a more creative way to get through it.
I think we need to work creatively together. I think that every parent would say that the placebo is really hard because waiting means time and time means loss of function. So that's our job together. I can't thank you all enough for joining us. This has been a great conversation. We're going to have many more of them. And I'll turn this back over to you, Eric.
Thank you, Pat. And thank you to everyone on the Solid team for joining us today and for running a little bit over. We really appreciate all of your insight and the additional feedback on your presentation today. And thank you, everyone, for joining us. This recording will be up on the PPMD site in the next week, so you can see it there if you want to get any more information or pass it along to anyone you know who wasn't able to be here today. So thank you, everyone, for joining us.